Without knowledge of the 4 different blood types, for much of human history, blood transfusions were a matter of pure luck, and most patients died when their bodies rejected the incompatible blood. Lance Steiner made his world-changing discovery of the different blood groups. His discovery made transfusions much safer, but one major drawback remained. As blood could not be stored outside the body without clotting, blood transfusions still required the donor and the recipient to lay side by side. In 1915, Lewison found the optimal concentration of the anticoagulant sodium citrate, which allowed blood to be stored safely for several days. Lewison's method of storing blood was adopted across the world, and blood transfusions between donors and patients who were great distances away from each other quickly became the norm. The pair's discovery made blood transfusions both safe and practical, and their work paved the way for the worldwide system of blood banks that we know today, which have so far saved over 1 billion lives.
Every 2 seconds, someone in the U.S. needs blood. In surgery, on cancer wards, and on America's battlefields, blood transfusions save lives. But in the U.S., demand often exceeds supply, and elsewhere, especially in the developing world, there's a real chance the blood could be contaminated with diseases like AIDS or hepatitis C. Enter Dr. Mark Turner, a cell biologist from Scotland who's received a multi-million-dollar research grant to make blood in his lab from human stem cells.
These cells are being generated from human embryonic stem cells, which themselves are generated from 3 to 5 day old human embryos.
Stem cells can be coaxed, theoretically, to grow into any human body part. Dr. Turner's team will try to make them grow into O-negative blood. If they're successful, the payoff is huge.
What would be phenomenal would be if we could create infection-free blood that is laboratory generated.